Okay, so we're starting again with question 4, which is about evolution as change of structure of DNA. So the first few are some multiple choice. Let's have a look. Now the first one says over here, put a cross in the box next to the term that completes the following statement. Each DNA consists of mononucleotides joined together by bonds between what? Okay, so first of all, we know that it's a deoxyribose sugar. And the other three BCD don't have it because it's DNA. DNA stands for deoxyribose nucleic acid. And so over here, the one which has deoxyribose is definitely true, and it also has a phosphate group because it's a nucleotide. If you can draw it out, I'll draw it out for you now. It's like a pentose sugar group, like this. And then it's just like this and this. That's one nucleotide, and then the phosphate group is going to attach to the house pentagon looking shape. So it's going to be this. Now this is also very simple. This is AS Unit 1 information. So if you don't know about this, you need to go back to your basics. Now put a cross in the box next to the term that completes the following statement. The mononucleotides in one DNA strand are joined together by phosphodiester bonds. Now this again is simple because you said that it's between a deoxyribose sugar and a phosphate group, and we know that every phosphate bond would start with the word phosphodiester because it's a diribonucleic acid. So this one was definitely simple. Okay, over here this one's a bit like when I did it wrong first. So it says we need to put their sample of DNA and we need to show how much percentage of bases are in a sample of DNA, which has 33% thymine. So we know that thymine it bonds to adenine, so that's going to be the same one. And we also know that uracil is not present in DNA, so that's going to be 0%. So what's left over here is cytosine and one other, what's it? Guanine. The C and G ones, which bind together. So if we put 33% plus 33%, we get 66%. If we minus that from 100, we get 34. And if we divide that by 2, we get 17. And thus the answer over here will be 17. I stupidly wrote 33% because I forgot that there's guanine as well. So we need to know that there are 4 bases and uracil is not present in DNA, so that's the major part over there. Then we're talking about species of rhinoceros. Over here, just by looking at the picture, I can tell that this question is about evolution, common ancestors, and how they speciated. That's definitely what's going to come in. So over here, first of all, is this calculation over here. We need to calculate how many times bigger a white rhinoceros is than a black rhinoceros. So what will we say? So they've given us the shoulder height and height. It has a shoulder height of 142.7. Okay, so they've given us like these two values. Okay, so how I did it was that we have to find how many times bigger the white one is. So, excuse me, we're gonna make a ratio. So white to black. White ratio black. Okay, so that's, I'll write a bit more clear because I feel like people don't understand my handwriting. White to black. Okay, so the white one is 1.5 times 1.8. We're multiplying those two values. Ratio 1.4 times 1.7. Now over here, just by looking at it, we can tell the white tortoise is bigger because it has bigger values, and these ones are smaller values in comparison. If we multiply it, we get 27 over here, and over here we have 23.8. After that, we're just going to divide it by 23.8. And over here we get 1, obviously. And over here, if we put it in a calculator, let me just do that. So we have 27 divided by 23.8, which gives us approximately 1.13 x. You don't need to write the x, so over here, how many times bigger is it? 1.13. So I'm just gonna write 1.13x bigger, just just to make it more fancier, and that's the answer. Okay, make sure you write it over here because they might be stupid. Your what's it called? The examiner, they might not give you the mark if you don't write it down. And that was just 2 marks. Now, suggest how these 2 species of rhinoceros evolved from their common ancestor. Okay, so definitely natural selection. That's the major point that we're talking about, is natural selection. I'm going to make a text box now. And that's a useless task for the time being. And let me make a text box here. So, due to natural selection, there was a change in DNA. Because the first question we're talking about DNA, so I'm just going back to it because I feel like we might need to put something about DNA there, because it needs to take part into the overall structure, so it has a flow to it, and I think I just want to keep that flow. So there's a change in DNA structure because of mutation. Okay. This may lead to a change in hip shape, because they said the hips changed over here. I read it somewhere that it has broad flat lips and has a pointed mouth, and they evolved from lip, not hip, lip, sorry, change in lip shape. And these alleles passed on to offspring, because their kids got that alleles, which led to a change in allele frequency over time. Okay. Then over time, it led to speciation, because they're 2 different species now. Speciation as they became reproductively isolated. Okay, why did I say that it's reproductively isolated? Because there's only 2 types of speciation that we know, which is sympatric and allopatric. Allopatric is geographical, and over here they're living in the same place. They evolved. They're both in Africa. They both live in Africa. They didn't mention that they're in different places, so it would be hard for me to assume that there was allopatric speciation. So because of they changed as they actually, I won't say that right now. I'll say that their food sources became different. As their food sources became different. Now, why would their food became different? Because you see, there's a change in their color. So because of that change in color, wait a second. I was just getting. Because of that change in color, I would say that they prey on different materials, because hiding from their faces, into because now they probably occupy different niches, I said because, oh god, I wrote became wrong. So because they now occupy different niches, they're gonna definitely have they're gonna eat different stuff, because they probably won't live in the same area. Because white rhinoceros would probably live in places where it could like blend into the background and then attack its prey that way, and the black one, the gray or black one, the black one, it will be able to do it in another way in like a places where there's like trees, maybe. So that's what I said. It will lead to speciation as their food sources became different, and this may lead to, again, it's not for sure because we don't know if they're, because we've only done like 2 types of speciation. So this may lead to sympatric speciation. The way that you write it has to be very specific. You need to make sure of that. And that's what I wrote here. Yeah, over here the main points which are gonna give you marks is mentioning natural selection. You can also mention selection pressure, but over here they didn't really talk about it. So we don't really know what selection pressure was there. So it's more easier to assume that it was natural selection. And then we can say change in lip shape, that's basically us applying it, and alleles will be passed to offsprings is another point, leading to change in frequency is another point, and over time it may lead to speciation. This whole thing is another point. And so we get the 4 marks. We did a bit more, but that's it. So the thing to take into account is over here, if you like read it, it's not a lot of, we're not taking up a lot of space, but we need to make sure we know what we're supposed to write, because we can fill it out with stuff that's irrelevant, but we need to make sure that it's and over here it suggests. So we don't know for sure because they didn't give us enough information to tell for sure why did they become 2 species. Over here they just says they have different food sources. The white one, it feeds on grass. This one eats shrubs. Excuse me. So that's why you can tell that they have different food sources. So that's why both of them can be found in the same region in Africa. So as I mentioned before, it can't be allopathic speciation because they live in Africa. Both of them, so they're not geographically isolated in any way. So they occupy different niches. As I mentioned that before, didn't I? They occupy different niches and have, because they have different food sources. And then this was already mentioned. The whole point over here, these are just the 2 marks that you can possibly get. And yeah, that was actually very simple because that's the only reason that it could actually be it. That's the only reason why it can be found in the same region, is because they occupy different niches. I told you, because one is white, one is black. They definitely they live in different, like, not that they live in different regions, but they live in different parts of the same region. You know, they prey on stuff differently, so that makes sense. And then we finished the question. So we have question 5 now, which is about infection and gut flora. So what is infection? So the term of infection, explaining the term infection, has come quite a few times in later questions, in later past papers as well, and it's always the same. We need to say that a pathogen enters a body, and not a body but a cell or a tissue of an organism, and then that's it. Pathogen enters and then into organism or cell. That's basically it. So I'll just write that down. Infection is when a pathogen enters an organism's, we need to be very organism's cell and adversely affects it. You don't really need to write that point. I just think I just feel like writing the first till here, writing till here is just like a bit, I don't know, I just don't really like the, it's very simple, but this is basically the thing that's going to earn you the 2 marks. You don't need to write the rest of it. You can say it harms it, but there's really no point, because it's a pathogen, so we know that it harms it. Now, if you need to explain how gut flora protects the body from infection, so obviously, if you've learned anything about gut flora, you know that it increases competition, because it basically is bacteria on your skin, in your gut. Sorry, that's skin flora. It's in your gut, and so how does it increases competition? Is we need to mention it. It increases competition, for in space, for space, as there is limited space. We can also say that it prevents growth of microorganisms, because it increases competition. I wrote growth wrong. Okay, microorganisms. We could also say that it releases toxins to kill microorganisms. So whichever one, actually there are 2 points, so you have to mention all. Also, it releases toxins to kill pathogens. There. You can say pathogens, microorganisms, bacteria, fungi, not fungi, bacteria, anything. And over here they gave us a graph, so we should be able to understand the graph first. It shows the diversity of gut flora if a person was recorded. The person then took a course of antibiotics for 7 days. The percentage of each type of bacteria in the gut flora was recorded for a period of 18 months. Each bacteria is represented by a different letter in the chart below. So, why is this not working? Oh well. So we have A, B, C, D, E, F, G, H, and I. And we can see that all of it's gone except G. So G is definitely resistant to that antibiotic. It's not working because it's still there. And then we have, then H comes back and I's come back. And there's this other one called J, which is not in the original set. So J is definitely a new one, and it's probably not coming because it was not already in it. It definitely came in for some other reason. What could be that other reason? I don't know. Well, I could I don't know if they're gonna ask us about it, but it is definitely confusing. H and I came in, and that's definitely because of they're consuming H and I. So it can be because of diet. It could be because they're taking some other not antibiotics, but they could be taking some other medications or supplements which has it, or it could just be contamination. Not contamination, to be fair. It's definitely coming out of diet. H and I. So that's what we know right now. So 12 to 18 months, they just came back. We need to explain the term antibiotics. So antibiotics, they inhibit. What do they inhibit? The growth of bacteria, and their chemicals. Now, the are chemicals part, it would not make sense definitely unless you gotta look at the marks. We can say that they are chemicals which inhibit the growth of bacteria. That's definitely true. It just didn't came to my mind, but it was present in the mark scheme, so that's where I actually lost a mark. So the biology mark scheme is, it's made in such a way that you're supposed to lose marks one way or another. You have to say that they're chemicals, which is kind of stupid, but ooh, we have a 6-mark question. But you need to be able to know it, so You have to be doing past papers to be able to understand that. Now, a 6-mark question, you know, the one with an asterisk is an essay style one, so you need to give it in like chain of reasonings and everything. So if the chain of reasoning is not present, it's not gonna work. So yeah, the question is we have to use information in the graph to describe the effect of this course of antibiotics on the diversity of gut flora, and then we need to suggest explanations for this effect. So basically everything that we said, we just need to mention it here in like in like a essay style, writing style, in an essay type answer. So over here we'll just look at it. So after 7 days, only one type of one type of bacteria is left. So I'm just gonna first of all state everything that's going on. Okay. So after 7 days, only one type of bacteria G is left from the 7 different types. I don't know why I'm writing that. It's supposed to be an SSL question, so okay. I just write them in points, so 6 different points. You don't really, for 6-mark questions, you don't need to write 6 different points, but you need to make sure that everything is just connected. So you have to actually write more. This may be, so why is G left? Because G may be resistant. This may be because G is resistant. Okay, so then what we're gonna say, after 12 months, H and I also appear in the gut flora again. Maybe because it's present. Again, I just said maybe because there might be some other factors at play we don't really know about. Maybe because it is present in diet. Present in the diet of the person. So I'm giving an explanation. I'm giving like a description and an explanation. Description and an explanation. So right now we've done 4 different points here. Then if you look at over here, G is still in majority. Even though after 18 months, there are more there are more bacteria, G is still in majority, is still present more. How much is this? There's more percentage of G. Okay, there's a higher percentage of there is a higher percentage of G. Now what may be the reason of that? Because it's resistant to be fair, it's probably mutated. So it's virus resistance because it was always present, regardless of the course. Regardless of the course of antibiotic. Maybe the antibiotics was not targeted toward it. Maybe the antibiotic was not targeted towards bacteria G. I didn't say bacterium because there's a higher, because it's in percentage, so obviously there's always more than one bacteria here, because it's in percentage. It would be probably like 0.0001% if there was only one bacterium. So I said bacteria. So again, even though it doesn't really matter, to be fair, but you have to use the correct singular, plural of every word. It's just me, probably. It doesn't really not matter that much though. So we didn't mention the new bacteria J. Okay. After 12 months, a new bacteria J appears. No, we don't really know. Okay. And then overall, just write just, even though I don't know if it's a mark. Bacteria A, B, C, D, E, F, H, and I are killed by the antibiotic. We can say inhibited. We can say completely, stop the growth. Yeah, and that's basically it. That's all I'm gonna write. So if you look at it, 1, 2, 1, 2, 3, 4, and then 5, 6, 7, and 8. I wrote 8 points, so hopefully it'll be enough for 6 marks. If you find anything else that you can tell from the graph, you can mention that as well. Question 6, we're done with this question again, and we're moving over to question 6. This is about volcano, definitely about succession, just by looking at it, because you can see over here, this was a climax community, and there was an eruption, and then it became bare rock, and then you can see some shrubs, something else growing on it still. So if I had to guess, I'd probably say, because over here there's a number of different plant species graph, and you can see that it was almost stagnant, because that's how you can tell it's a climax community, because it's reached that point, then it immediately drops because of the eruption, and then it slowly increases again, and that's the primary succession. So I should write that down for yourself. You can just write that down over here. That's primary succession. Ooh, I can't write. Okay, that's primary succession. There you go. Okay, we have an MCQ over here. The increase in the number of different plant species in Mount St. Helens or fall area following the eruption is an example of deforestation. That's stupid. Evolution, no. Natural selection, no. And succession, here we are. I already mentioned it before, so it shouldn't be a problem. Now, using the information in the photographs and the graph, explain the changes in the number of different pest species in Mount St. Helens area. So, again, over here, we'd explain the changes. So this is because of primary succession. This is because of primary succession. Because of the lava, no organism could grow. And all the plant species died. Okay, and then after the lava cooled down, what happened was that microorganisms come first. So microorganisms came, started to arrive, so came and respired to increase our fertility, to increase soil fertility. We can also mention the bare rock. The bare rock eroded, and so that pioneer species, you should mention the fancy words, pioneer species like what could that be? Moss and lichens. That's the ones that we learned about. So moss and lichens are able to grow. And then we have bushes, then we have, so the number of plant species grows slowly, number of plants increase, and so does species as bushes and other small shrubs start to grow. There. Okay, there we are. I spelled increases wrong. Increases. Excuse me. There we are. Suggest how, okay, we already got a climax community question. Suggest how further changes in mountain tolerance area could lead to development of a climate community. So they already had one before. So the increasing, what are the further changes that could be? So they could increase the number of animals there, because as we know right now, there are plants bring in, but you know, animals can't just randomly come up, so they can introduce animals species to the area. I just don't want to write mountain intelligence because it's just very odd. To the area. As time goes as time in time taller plants would come in, taller plants will grow, and the soil will become deeper because of increase of microorganisms, because of increase in, and not only because of microorganisms, to be fair, we're also going to say that the plants will die. Plants which die will increase the fertility of the soil, will moreover increase fertility of the soil, and yeah, and soon there will be an ecosystem similar to what was before, the eruption. There we go. There is, that's 3 marks. So introduce animals to stage 1 point, taller plants is another point, soil become deeper is another point, and similar to what it was before the eruption is 1 point as well. Then we're talking about GPP and NPP. So first of all, let's just review our knowledge about it. GPP is gross primary productivity and NPP is net primary productivity. So if you remember the formula, it is that over here, let's look. We need to know, okay, we need to show which line is which. So what is more, GPP or NPP? Definitely GPP, because the formula for GPP is, oh gosh, ignore my handwriting, because GPP equals NPP minus R. Okay, so Q is definitely respiration, so anything which says Q is R, Q is R. So Q is GPP over here, that's definitely wrong. And that's definitely wrong. And then the answer is C, because there's supposed to be, this is supposed to be GPP. and that's supposed to be NPP. There. And that's it. That's the question. Oh, we're done. Then we have question 7, which is about fungus and its multiplying. So binary fission. They do grow asexual reproduction. I don't really know what the question is about. Suggest how properties of cell membrane enable the yeast to form a bud. I don't understand. And then cell cycle, okay. And how temperature affects asexual reproduction. So basically everything here. Okay, so yeast is a single-celled fungus. It can reproduce asexually in a process called budding. When the bud is big enough, it separates from the original yeast cell. Yeast can be grown in a culture containing all the nutrients needed for growth. Soil samples of the culture can be removed and the yeast observed using a light microscope. The photograph below shows the yeast budding as seen using a light microscope. So you can see over here that their cell membranes are fused together. So we can say that it's fluid. The cell membrane is fluid because you can see it's changing shape whenever it's budding. So its shape can be changed. And yeah, we just need to explain that. So it's very similar to what happens when there's like in the Golgi apparatus in the human body, and like this is a human cell, how it just pinches off and separates. That's basically, it's almost similar to that. So I would say the cell membrane is fluid. This question is, though I don't think we've actually studied that much about yeast cells, so we have to definitely make it up over here. We need to look at it carefully to be able to tell, because I don't think the knowledge about it was given to us beforehand. So it can change shape to multiply and bud. They can pinch off. They can pinch off. I'm using the same terms as I used when I was talking about, if I was talking about the Golgi apparatus and how, you know, a new modified protein is getting out. It's mostly the same words that I'm using. Pinch off and separate. I'm just making it more in terms of yeast cell. So it can change shape and multiply and bud. Yeah. So yeah, that's basically it here. Yeah, that's the 2 marks. It's fluid, it can distort, and then it pinches off and separates. Then we need to explain the role of cell cycle in yeast budding. Cell cycle is interphase, anaphase, prophase, all of that. We need to explain how DNA is synthesized here, how means that it's gonna have, because it's budding, so the yeast cells are gonna be genetically identical, and then there's gonna be cell division, and then on which phase, actually it's interphase, where the organelles are being made. So we're gonna start off. DNA is synthesized in S phase. Okay, then synthesis of organelles, because it's a single cell, so it's not gonna have organs. It's gonna be organelles because they're tinier. It's gonna be in interphase. This again is information that you should definitely have before doing past papers. It's just concepts. And the bud that grows will be genetically identical to parent cell, and then cell division causes cells to form. This is just information that you should know. And yeah, that's basically it. That's the 4 marks. There's probably going to be an extra mark, but I don't know what's for. Like, this is going to give us 4 marks, but I'm pretty sure there has to be an extra mark. In 4 mark questions, there's always one other point, like an extra point. Like, if you don't write this, you can write that. I don't know what that is though. It has to do with watts. Yeah, there has to be one. I don't really know, but this is gonna give us 4 marks for sure. This is a asterisk question, so we need to be careful about how we write it. So we need to suggest an investigation that could be carried out to study the effects of temperature on the rate of asexual reproduction. Okay, so I'm gonna be writing like a whole thing. You probably would have done this experiment in school. We've done a yeast cell one. So in a sterile environment, agar dishes, jellies, agar jellies can be used to create a culture. Again, the language over here matters a lot. To create a culture of yeast cells. So we have to count the number of yeast cells using a microscope. And put it in 6 different dishes, different agar dishes. Okay, then put them in different temperatures. Place them in different temperatures, and then I'm just gonna put some temperatures in 20, 30, 40, 50, and 60 degrees Celsius. How do you make a degree Celsius? I don't know. So I'm just gonna do this. Let's see. 50 to 60 degrees Celsius. Then we're gonna be make sure all other variables are constant. But now we're gonna have to talk about what other variables are there. So I'm gonna start off with this includes glucose content, because obviously we want them to respire, so there has to be some food that you need to put in it. If you've done the experiment, you will know this includes glucose content, not constant, content. Shh. This includes glucose content and size of dish, and then because obviously you need to make sure that there's the same space for it. Then after a week or so, or it can be 24 hours. You don't want them to go for so long. After 4 hours, then the number of yeast cells should be counted again. And we have to repeat the experiment to get more reliable results. All the experiments questions follow like a strict procedure, so you should be able to do this question without any actual issues with it. I'll just add aseptic technique again. Use aseptic technique because I mentioned sterile environment in the first, so I have to mention it. Use aseptic techniques like wearing gloves and keeping all the equipment sterile. And taking as little time as possible. To make this culture, because if the more you delay it, the more it's going to take, the more risk of contamination. To take as much as possible to make the culture to reduce contamination. There's I've never done an experiment where there hasn't been some kind of contamination. I don't know, I'm just really bad at it. And that's to make the experiment valid. and calculate rate at the end. Calculate rate to be able to study effects, to be able to compare results. There we go. That's it. That's, again, it's a very simple question. If you know what you're talking about, it's not going to take you a lot of time. It's very simple, and you should be able to get full 5 marks in this one. And then we're going to do question 8 and that's question 8. We're going to do question 8 later. This is basically because it's already been taking us too much, like 45 minutes already, so we will be able to do that later.